Okay, so this lecture is going to be on the protozoa. These are your blood protozoans that we're going to talk about. The information is found in your textbook, chapter 28, Textbook of Diagnostic Microbiology, 5th edition. These protozoa fall under the phylum Apicomplexa. These are blood and tissue parasites. As the name mentions, they have a complex life cycle, both sexual and asexual. Some of these that we'll talk about require vectors, insect vectors. Some of them, the human is the definitive host in which there's sexual reproduction, and some of them are intermediate host in which there's asexual reproduction. We will talk about plasmodiums. These are your malarias, babesia, toxoplasma, cryptosporidium, isopora, and cyclospora. In this lecture today, we're going to talk primarily about Plasmodium species. These are your malarias once again. We'll deal with Plasmodium vivax, Ovalii, malariae, falciparum, and Knowlesii. Knowlesii is a zoonotic, and it's usually found in primates, but we have recently learned that we see it in co-infections with other Plasmodium species in humans. Plasmodium causes malaria, and what malaria is a bout of fever and chills, and these are called paroxysms. You'll see splenomegaly, which is an enlargement of your spleen, and you'll also exhibit anemias. You'll see asexual reproduction in humans. There's 2 cycles with that. An exoerythrotic phase, which just means outside of the red cell. This occurs in the liver, and then you have an erythritic phase, which is within the red cells. You also have the sexual reproduction in the definitive host, and this typically occurs in the females and the female Anopheles mosquito. You will see sexual forms of these parasites in humans, and we'll talk about that in a moment. Your infective stage will be what's called sporozoites, and these are injected into the bloodstream by the mosquito. The diagnostic stage will be found doing thick and thin blood smears, and we're going to look for characteristic forms of the parasite. The morphological forms we're going to look at with each species of Plasmodium. We'll talk about a ring form, and this is just an early trophozoite. We'll talk about the trophozoite, an immature schizont, a mature schizont, and these contain merozoites. And then we'll also talk about microgametocytes, which are your male, and macrogametocytes, which are your female. The ring form is usually seen as an early trophozoite. It invades the red blood cell. It's going to contain a blue cytoplasmic circle, and a red chromatin dot will connect this cytoplasmic circle at the nucleus, and inside the space is a vacuole. It kind of looks like a diamond ring. These are pictures of that, and the one on the left, you can see the single ring form in each red blood cell. One on the top right have double ring forms. So how many ring forms there are is characteristic of species of Plasmodiums. The mature trophozoite will start to lose this ring appearance. You'll see an increase in the cytoplasm. The chromatin dot will remain, and you'll see remnants of the ring, and it will occupy more space within the red cell. And this is a picture of that, where you can start to see some of these these ring forms falling apart. Okay, the schizonts are multinucleated stage. You'll have an immature where chromatin replication is occurring. And then as they mature, they'll start containing merozoites. Merozoites are just that chromatin mass that's replicated, surrounded by, each one of them would be surrounded by cytoplasm. The cell will rupture and release these merozoites, and this is linked to when symptoms start to appear. The number of merozoites that's released is species specific. And this is a picture of a schizont and a red blood cell. And those are individual merozoites. Then also you will see sexual forms. The microgametocyte is the male. You got a pale blue cytoplasm and diffuse chromatin mass, or you'll have large chromatin surrounded by a colorless halo. They're typically round except for falciparum, which they're crescent shaped. The macrogametocyte is the female. It's well-defined compact chromatin, dark pink. It's round to oval, once again, except for falciparum, which is crescent shape. You can see the gametocytes within the red blood cells, or you can see them free-flowing within the blood. Here's a gametocyte within the red blood cell. And here's a gametocyte that's free-flowing outside of the red blood cell. This happens to be falciparum, that crescent shape. How do we diagnose malaria? You want to get a good travel history. You want to take note of the paroxysm, the fever and chills alternating. We do a peripheral blood smear. Using EDTA blood is the best. We make a thick smear, which is on the top to the right. And we use that for screening purposes. And then we do a thin smear, and we do that to speciate. And you want to look at about 200 to 300 fields. You want to collect the blood prior to a fever, and you need to do more than one per 36 hours because these are cyclic. Acridine orange, which if you remember is the fluorescent stain that you can use in microbiology, it just tells you whether or not there's an organism there. You can't use this to speciate. You can do serological testing. They actually have some rapid tests out now that use monoclonal antibodies. They also have a couple of tests that do species-specific lactate dehydrogenase. The problem with the serological method is sensitivity limitations. They do have PCR, DNA molecular tests out there now. The gold standard still is thick and thin blood smears. Okay, so the life cycle, we're just mainly interested in what goes on in humans. So you got the asexual stage in humans where the sporozoite is transmitted by the female Anopheles mosquito. There's two cycles of this. There's an exoarthritic cycle, and this is just outside of the red blood cells, and it happens to occur in hepatic cells, so this is going on in your liver. The schizozony form is of the asexual reproduction. This is where your merozoites are produced and released into the bloodstream, and then they'll invade red blood cells. And the erythrocyte cycle, so once in the red blood cells, you'll go through your ring form, through your trophozoite, to your schizosomes, which rupture, releasing those merozoites again. You'll also have those macrogametocytes and macrogametocytes. We see those when they're transmitted to the mosquito during feeding. You can also see those within the red blood cell and outside of the red blood cell. Here's a depiction of the cycle, and we're mainly just interested in what happens in the human. The gametocytes that you see in the mosquito, we're not going to worry about, but you want to mainly know what goes on in the human. Here's CDC depiction of it. Alright, so let's talk about dormant schizogony. This is basically a resting stage that occurs in the liver, and it can result in a relapse within 1 to 5 years after being exposed. It's been noted in Vivax. They also thought they had documented valley cases of this, but now they're beginning to think that this is not correct. So how is malaria transmitted? The vector is Anopheles mosquito, typically the female. We've seen it occur in blood transfusions, sharing of needles, bone marrow transplants. There's also a congenital component because it's been found in the placenta, and this can compromise fetal development. Treatment is typically classified by the stage that the drug attacks. The problem with treatment nowadays is like everything else, we're starting to see drug-resistant malarias. Falciparum is one of the biggest ones we're seeing resistance in. There are some immunity concerns. Their complete immunity doesn't exist. If you get it, you can get it again, even though you've been treated. Individuals in endemic areas, though, will develop antibodies, but it's only against the asexual stage. This is why some of the ELISA immunoserology tests don't work. Individuals with certain hemoglobinopathies will have a limited immunity to malaria. Sickle cell disease, hemoglobin C disease, glucose 6 phosphate dehydrogenase deficiency. And then there's a weird immunity with Duffy antigen. It seems that there's a receptor for Plasmodium vivax and knowlesii on the Duffy antigen. Okay, so let's start talking about the individual species. Plasmodium vivax is known as benign tertiary malaria. It only affects reticulocytes. Remember, reticulocytes are your immature red blood cells, so they're going to be enlarged. You'll see Schuffner dots, which is known as stippling except in the early ring forms you won't. The trophozoite will be an amoeboid appearance. And the mature schizonts contain 12 to 24 merozoites with an average of 16. And here's a depiction of all those forms. You can see, you want to notice that the red blood cells are large because they're reticulocytes. You see the Schuffner dots. One of the things about invading just reticulocytes is that it reduces the number of red cells that the malaria can invade. Plasmodium vivax is the most widely distributed. We see it in the tropics, the subtropics, and temperate zones. It's called a tertiary malaria because eventually it settles into a 48-hour life cycle, and then at the start of 72 hours it invades a new group of red blood cells. If left untreated, the infection can last for several years. And remember, this is one of the ones that has the dormant phase in the liver. Plasmodium ovale, this invades reticulocytes also, so it invades the red blood cells. It's oval in shape. You may see finger-like projections on the trophozoites. You also will see what looks like comet forms. Schuffner dots are present, and the mature schizonts contain 8 merozoites. These are the pictures of ovale. Once again, it's only in the reticulocytes, so those are the large immature red cells. So it's only invading about 5% of the red cell population. It's located in Africa, the Middle East, Papua New Guinea. It's similar to vivax but less severe. It's a tertiary malaria, meaning that it life cycles every 48 hours, and then on the 72-hour mark it invades a new set of red blood cells. And they're not sure about the dormant phase in this one. Plasmodium malariae is a quartan malaria. It infects normal red cells, so the population that it infects are much greater than just reticulocytes. It has no Schuffner dots. The trophozoite is in a band form. You'll find many trophozoites and schizonts in the blood. Mature schizonts contain about 6 to 12 merozoites with an average of 8. Here are the forms of that. One of the main forms is you want to notice the band form of the trophozoite. That's a key characteristic. Red cells will be normal in size, not enlarged. Plasmodium malariae is a quartan malaria, meaning that it has a life cycle every 72 hours with a new invasion of red blood cells on the 4th day. If left untreated, the infection can last for several years. This Plasmodium has a latent period with a persisting low-grade parasitemia, so it's different than the dormant phase that we've seen in the liver and it's in the other phases. You'll see proteinuria, which is characteristic for malariae, and they think it may be associated with nephrotic syndrome. They also see an antigen-antibody complex in the glomeruli, they'll see compositions of that deposited in the glomeruli. Okay, so Plasmodium falciparum is called malignant tertiary malaria. It affects normal red cells of all ages. So it's probably the most populated red blood cell malaria. There are multiple ring forms. It has a crescent or banana-shaped gametocytes. There are no schizonts in the blood. It has what's called Maurer dots, which are red comma-shaped. And the mature schizonts contain 24 merozoites, and because they're not in the blood, you'll see these in the tissue, like in organs of the liver. These are the different forms of falciparum. They're found in tropical zones. It's called malignant tertiary malaria. The life cycle is every 36 to 48 hours. It's kind of asynchronous. You want to associate black water fever because this is hemoglobinuria, which is where you get the black water from. Can lead to renal failure. Falciparums have a tendency to cause the red cells to be sticky, and cell debris can clog up the capillaries, especially in the brain, and lead to death. Plasmodium knowlesii is known as simian malaria. It affects all ages of red blood cells. You'll see multiple ring forms up to 3. There are no Schuffner dots. The mature schizonts contain 16 merozoites, but no rosettes. And you usually see this with a mixed infection, and it can be fatal. This we haven't studied much until recently. It was primarily a zoonotic infection seen in primates. And then here is a chart given the key characteristics of the malarias minus the knowlesii.